Okay, hey, good morning. How are you guys? Let me change this. Good morning. How's everyone feeling today? I hope feeling pretty good. All right, let's see where we're at here. Wow, we've come so far. We've come so far in the plant kingdom. Let's pull this up. Make it big. Oops. Stop share. Hold on. Sorry, I'm having an issue. 1 second. Oh, get out of there. There we go. Okay, share screen. Yes, do this. Go there and make it big. Great. And fix this thing. Pull it up to the side. Oh boy. Oh man. There we go. Okay, finally. Woo. Cool. So good morning. I hope your week is going well. It's Thursday, yay! So tomorrow's Friday. You guys have lab, and then weekend. So just moving through the plant kingdom. I will say today is kind of a review because on Tuesday you guys have your exam. So today serves pretty much as a review. I should point out, like on the syllabus and stuff, it's, the exam covers lectures 1 through 8, right? So from the very first intro class all the way through the horse tails. But this is technically lecture 11, but lecture 11 is like a review. So feel free. In fact, you should look at these slides too, because it kind of helps organize all of the information, not all, a good amount of the information, or at least the core concepts of the lectures 1 through 8. So yeah, that's what really today we're going to focus on, because is the core concepts. Because sometimes we look, when we're going through each lecture, there's a lot of details about a given plant species and how it pertains to other plant species, but we go into real details about the structures and whatnot. And sometimes it kind of lose sight of that bigger picture and the core concepts of this class. So today we're going to talk about that. So what have we even learned so far in all of these classes? Well, we've learned that there's a lot of plant species, right? Almost, or probably more than 400,000, but at least more than 390,000 that we know of so far. And we know that the majority of the plants that are on Earth that we know of are flowering plants, right? Are flowering plants, which we haven't even talked about yet. We talk about flowering plants at the end of this course. So, but that, whoa, all right. Make sure you guys mute. Why is this not, here we go. Yeah. So the majority of the plants are the flowering plants, but we haven't even talked about so far. But we have talked about some kind of steps that have occurred so far in the evolution of plants that ultimately will lead to the evolution of the flower, So here are some of those things that we've already talked about in the class that ultimately lead to the flower. So number one is all plants have alternation of generations. However, when we go from non-vascular plants, which have the gametophyte generation as dominant, to the vascular plants, which have the sporophyte generation as dominant, we have a shift, right? We have a shift in dominance. And so that's super important in the evolutionary history of plants, but also in eventually leading to the flowering plants. We also have seen as we go through an increased complexity of the sporophyte plants. So for example, way back when we were talking about the mosses, Polytrichum moss, for example, that sporophyte plant was like this big, it's itty bitty. And that sporophyte plant had to be attached to the gametophyte plant, which was much larger than that little sporophyte plant. And that sporophyte plant wasn't photosynthetic. It had a foot, a seta, and a capsule. It didn't have any roots, didn't have any stem, didn't have any leaves, wasn't able to do any sort of thing like gas exchange, right? Then you get to the liverwort sporophyte plant. Well, you still have a foot, a seta, and a capsule, but now it's itty bitty, it's even more itty bitty, and it's hanging upside down off of a new structure. Then you get to the hornwort plants. Sporophyte plant looks like a horn, still really itty bitty. Still doesn't have roots, still doesn't have anything like that, just has a foot, an intercalary meristem, and then the capsule, which looks like a horn. But at least then, now that sporophyte plant has stomatal apparatus, so you're getting a little bit more complex, right? Then we get into the vascular plants, and then, whoa, look at how complex we get. By the time we get to the lycopods, right, Lycopodium and Selaginella, the sporophyte plant has roots, has a stem, has leaves, etc., right? So that's what I mean by that increased complexity of the sporophyte plant. We also have a size reduction in the gametophyte plant. As I already said, in those non-vascular plants, the gametophyte plant's dominant. So when you see those plants, that's basically what you're seeing, right? Green fluffy in the case of the mosses, liverwort, and the hornwort kind of look like a flat green thing, right? Smaller than the mosses, still bigger than their respective sporophyte plants. But again, once you get to the vascular plants, that gametophyte plant is even smaller than it was. In the cases of Lycopodium, it could either be a little green blob or something that kind of looks like a carrot. Selaginella is even more small gametophyte plants because Selaginella is heterosporous. So the gametophyte plant has to be inside of a spore. So these are just some reminder terms, right? By now, these should be old news. By now, we should know what a sporophyte plant is and a gametophyte plant without even thinking about it, but just in case. And I just want to point this out because some of you still are getting these wrong on assignments. The sporophyte plant makes spores, guys. It makes spores. And those spores grow into the gametophyte plant. The spores grow into the gametophyte plant. The gametophyte plant makes gametes, egg and sperm. Gametes, egg and sperm. Okay. This is something we covered in the second class, this idea of alternation of generations, right? All plants have alternation of generations. Alternation of generations is a unique thing to the plants. Animals don't have alternation of generations. Even though we use that example of, a butterfly or a caterpillar turning into a butterfly, that's not alternation of generations because both of those things are diploid. Alternation of generations specifically refers to two organisms, one of which is a haploid organism, one of which is a diploid organism that are each part of a single life cycle, right? So we don't need to go through this step by step, but I like this slide a lot because since all plants do alternation of generations, it doesn't matter what plant you're talking about. You could be talking about a horsetail, you could be talking about a sphagnum moss, and then later on, you could be talking about a flowering plant when we get after, later on in this course. And it doesn't matter what plant you're talking about because these things, each of these things on this slide are going to be a component of their alternation of generations. Doesn't matter who you're talking What else can I point out here? I've color-coded this slide to show you things that are diploid as versus haploid, right? So all of the stuff on the left side of the slide that's kind of purplish blue are diploid things, and AKA part of the sporophyte plant. Part of the sporophyte plant, or the sporophyte plant itself, right? And then the things on the right side of this slide that are in orange are all haploid things, the result of meiosis and cytokinesis. And those things either grow into the gametophyte plant, are the gametophyte plant, or are things on the gametophyte plant, like the Antheridium archegonium, or are things that the gametophyte plant makes, right? Egg and sperm. Okay. Right. This is so when you're looking at the slides later. So every plant does this alternation of generation cycle, every single one. But as we move forward in evolutionary time, from the earliest plants to plants that have evolved more recently, we see these same trends of a more complex sporophyte plant and a more reduced gametophyte plant. All right, what does this say? Right, that's what I just said. Right, this is again, so that when you guys are looking at this, right, in the primitive plants, the non-vascular plants, it's the gametophyte generation that's dominant. Higher evolved plants, sporophyte generation is dominant. Dominant means exists most of the time, right? The most, like if you were to walk and see one of these plants, if you saw a moss, a liverwort, or a hornwort, you would be seeing the gametophyte generation with your eyes, with maybe the sporophyte generation coming out of it, maybe, sometimes. However, if you're seeing a lycopod or a horsetail or ferns and other things we'll cover later in this course, when you see them with your eye, you are seeing the sporophyte generation, and often you can't even see the gametophyte generation anymore unless you use something like a dissecting microscope. They're that small. All right. So why did plants evolve in this way? Like, why did we have non-vascular plants and then ultimately vascular plants? Why did we have gametophyte generation dominant and then later sporophyte generation dominant? Why do we need a more complex sporophyte generation to begin with, right? Why? I mean, mosses, liverworts, and hornworts, they don't have a complex sporophyte generation, and they do just fine for what they want, for what they need to do, right? So why did we end up here now with a more complex sporophyte plant? Well, remember that plants originated in the ocean, And if you live in the ocean, there's certain things that you don't really need. Like, so first of all, you definitely don't need roots. and you definitely, and if you're surrounded by water, you really don't need effective ways of transporting water. Like, you don't need xylem and phloem to transport things in order to survive, because you're surrounded by water. So you can just use the process of osmosis and diffusion to pull in water and other minerals, and then use the sun to undergo photosynthesis and make your own food, right? So you don't need to be a complex sporophyte plant if you're living in the water. Plants and green algae are based on DNA evidence, and based on DNA evidence are the closest living relatives to one another, which tells us as scientists that they share a common ancestor, or are derived from a common ancestor. And they share certain characteristics that reaffirm this, right? I think, for example, one thing they share is they store their food as starch, right? They have chlorophyll pigments and other things that they share. Eventually, plants, though, arrive on land. And the reason why is because there is land, first of all, And it's a whole new way, a whole new part of the earth to colonize and get resources there. But in order for plants to move from an aquatic existence to a primarily terrestrial existence on land, there has to be some evolutionary change, because what worked well in the water is not going to work on land. It just isn't. It just isn't. Right, that's what I just said, right? Right? The first land plants appear. This is a new niche space, so meaning like an ecological niche. Niche is a term that means like a area to move into, an area to move into. But like I said, living on land, way different than living in the ocean. And not only that, land isn't all the same, right? You have tropical land, you have desert land, you have Arctic land, you have mountainous land, temperate forest or temperate land. No forests yet, right? When plants first arrive, no forests yet. So all these different environments are on land. So in order to survive in a given environment, you're going to have to kind of evolve certain structures for it to work. And some structures will be just fine in certain environments, and other environments will require additional structures. Right, see, did fine. So here you go. First land plants did fine with a dominant gametophyte that had certain features. So these are those first land plants, those AKA non-vascular plants with their dominant gametophyte. What did those first land plants have? They had a cuticle. Cuticle is part of the epidermis that prevents you from drying out. We as terrestrial organisms, as humans, also have a cuticle on our skin that helps us not dry out, because we're mostly made of water. What else did the first land plants have with their dominant gametophyte that allowed them to be on land? Well, they had, so gametangia and sporangia. Don't get scared about gametangia. Don't get scared about tangia. It's just saying gamete making things and spore making things. Gamete making things, aka gametophyte, and spore making things, aka sporophyte, aka sporangia. Well, aka sporophyte with sporangia on it. That are multicellular. Actually, I should be more clear. Gametangia means organs that make gametes. So in the case here, organs that make gametes are your Antheridian archegonia. And sporangia is an organ that makes spores. So this is saying those first land plants, right, had Antheridian archegonia that were multicellular and sporangia that were multicellular, which is not what plants had when they lived in the ocean or the water. So that was a big leap, a multicellular sex organs and multicellular spore-producing organ. And the other thing they had is, because their archegonium is now multicellular, once you have the fusion of egg and sperm, the zygote is better protected than it was before, because now the zygote develops inside of that multicellular gametangia, in this case, an archegonium. So these are things that the first land plants had and allowed them to survive on land. But you can't live everywhere with just these things. You can't live everywhere with just these things. It's not enough. It's not enough for every environment. Got a question in the chat box, which I don't know why it makes me chat. Do I have a question in the chat box?
I'm sorry, Professor, you didn't have to come out. I could have just asked.
What's the question, hon?
Where was the zygote in the older plants?
Great question. So in the plants that lived in the water, the zygote would have been just free floating in the water. So in the water environment, egg and sperm would have both been just released into the water. And eventually they would have come together. But then the zygote itself would literally just be floating around in the water with no protection at all, which worked great in the water because it's almost, it's not going to dry out. It can pull in water on its own. But if you're living on land, you can't just like throw an egg out and throw a sperm out and hope they're going to fuse together. And if they did fuse together, you can't just have a 2N zygote just like out. It wouldn't work on land. Great question. Great question. Yeah, anyone else who has questions, feel free to just speak out, especially if you write something in the chat box and I don't, sometimes my chat box acts funny and I can't see it all the time, which is annoying. Anyways, yes. So these are things here that the first land plants had, but that doesn't work everywhere. And if anything, having a cuticle and the multicellular gamma-tangent-sporangia and a protected zygote is great. It's especially great if you live in a wet and a humid place, right? Because remember, these first land plants still don't have any sort of vasculature. But for plants to occupy more niches, they're going to need more complexity. And that's because they're going to need better ways to acquire resources, right? So it says here they need to evolve structures that can acquire resources more efficiently because they're going to need to spread their progeny to further distances. So progeny means offspring. And this is kind of similar to what I was just talking about zygote being floating in water. If you're a zygote, you're an offspring, right? And if you're floating in water, you can travel in the water. You can just like float away, go somewhere else. But once you're on land, you can't just float away anymore. So you have to have other ways that are going to be better at like spreading your offspring out further distances.
Professor?
Yes.
So when the egg and the sperm meet and they like float away together, it's not on the gametophyte plant anymore?
In the water?
Yes.
Yeah, so in the water, the gametophyte plant would have released the egg in addition to releasing the sperm. In the water.
Oh, okay. Those non-vascular plants, that was on land.
Yes, the non-vascular plants that we're talking about here, for these early land plants, those are not releasing the egg anymore because now they have a multicellular archegonia and a multicellular antheridia, and the egg stays inside of that multicellular archegonia now.
I'm following you. Thank you.
No problem. I'm happy to clarify. If you have that question, I'm sure other people have that question too. They're just maybe not as brave to speak out and ask. So keep them coming if you have any more things that need clarification. Yeah, so spread progeny to further distances. That's kind of a way of saying that we need something to move our gametophyte plants around better. And so, spoiler alert, it's the spore, right? It's the spore. Once you have spores that can fly away on the wind and then land and grow into a gametophyte plant, which will then have your sex organs, that helps you spread your progeny further distances. And then overall, you just need better protection for the egg. Better protection for the egg. If you guys also remember, in lecture 4, we kind of talked about the important characteristics of non-vascular plants versus vascular plants and like the pros and cons of each type of plant. So you could refer back to lecture 4 for more information about that. All right, so we're on land. We have non-vascular plants. They have the gametophyte generation dominant, right? And over time, in order to achieve, remember, we want to achieve, these are the things this is the things we want to achieve if we're going to occupy many niches, right? We need to achieve these things. Evolve structures that can acquire resources more efficiently. That means vasculature. Spread progeny to further distances. That's talking about spores, which will grow into gametophyte plants. And then better protection for the egg. We'll get there. We'll get there later. So, and if we're going to achieve those things, then we need to have a more complex a more complex sporophyte plant if we're going to achieve those things. So first, in these earliest land plants, right, these non-vascular plants, I already said it, but just to reiterate it, you have a small sporophyte plant that is completely dependent on the gametophyte plant, right? Completely dependent. Where's the sporophyte plant on the moss? It's this, and it's attached to the green fluffy, which is the gametophyte plant. Where's the sporophyte plant on the liverwort? Hanging upside down off of this new structure that the liverworts have. Where is the sporophyte plant on the hornwort? It's the horn, right? It's the horn. But they're all small and they're all attached to the gametophyte plant and dependent on gametophyte plant. They cannot survive on their own. This is just a reminder, right? These are two types of mosses we looked at: Polytrichum moss. Polytrichum moss sporophyte plant has a foot, has a seta, has a capsule. Sphagnum moss has, we'll start from the top here because it's easier, a capsule. And in here it has a foot. And that's it. Remember that the part of this little stalk thing that elevates the sporophyte plant is actually part of the gametophyte plant, right? And that's called, what is that thing called? What's the stalk called that elevates the sporophyte plant in the sphagnum? I sang a Phil Collins song to to remind you. What is it? Yes, yes, pseudopodium, right? Pseudopodium. Yes. Thank you. Okay. So we're talking about increased complexity of sporophyte plant. That's what we're talking about here. So on the previous slide, we saw the mosses. Here's liverworts and hornworts. Here we go. Here's liverworts and hornworts. Where's the sporophyte plant on liverworts? This whole thing? No, not that whole thing. Just this. That's the sporophyte plant. Just this. Just this. These are sporophyte plants hanging down. Look. Sporophyte plant, hanging upside down like a bat, right? On your liverworts. All the other structures you see there are the gametophyte plant. And then where is the sporophyte plant on this hornwort? It's this horn, right? It's this horn. So from using the naked eye, maybe it doesn't look like we're getting more complex, you know, because these things are all very small and they're all very green. Or sorry, they're all very small. And importantly, they are not all green because look, these sporophyte plants are not green, right? The one on the left is kind of like a tan golden color. The one on the right, it almost kind of looks like a Kalamata olive or something, right? It's kind of purpley brown. That's not green. So here we know for sure the sporophyte plants are not photosynthetic. We're talking about complexity, increased complexity. If you look at the you look at the liverwort on the left, that yellow thing hanging down that I just drew on before, that's the sporophyte plant. That's not green, right? So that's not photosynthetic. But by the time you get to the hornworts, oh, hey, oops, what do you know? What do you know? A greenhorn, right? So the sporophyte plant is officially photosynthetic. So that's increased complexity right there, even just that. All right. But still, to be fair, right, the liverworts, hornworts, and the mosses, their sporophyte plant is still pretty basic, right? It's not super complex, but even though we do see a slight trend towards becoming more complex, but we really won't see the complexity of the sporophyte plant really take off until we get to the vascular plants, which is what we will do here, right? Oh, here you go. This is one of those things from that lecture 4 that I just talked about that you could review, but this is part of it, right? Important characteristics of a non-vascular plant, important characteristics of a vascular plant. What do you notice? They're basically reversed. They're basically reversed. And this was a question that we had in one of the assignments, and most of you got this right. Some of you overthought it a little bit, but look, non-vascular plant has no vascular tissue. Vascular plant has vascular tissue. Non-vascular plant has free-living gametophytes that are dominant. Vascular plant has free-living sporophytes that are dominant. And non-vascular plants have a sporophyte attached to and dependent on the gametophyte. In other words, a reduced sporophyte plant. Vascular plants have a reduced gametophyte plant, right? So they're kind of opposites. But yep, now we are in the dominant vascular plants. A switch in dominance. Gametophyte generation no longer dominant. Sporophyte generation dominant. Hence, going to become more complex, right? We're talking about increased complexity of sporophyte plant. We're going to get more complex sporophyte plants. Here we see a bunch of our friends of these earliest non-vascular plants, right? Like when we saw this before, we didn't know enough to like talk about them, but look, here's your Cooksonia, right? In the back here, you see some sporophyte plants that start to have lateral sporangia, right? Look at here, this is Lycopodium. These are earliest vascular plants now getting established on land. But they need to have more complexity. So evolved structures that acquire resources more efficiently, that means vasculature. It needs vasculature, right? And so here you go. We looked at these before. Aglaophyton is our, here I'll change colors. This is from the lecture 5. This is from lecture 5. We saw Aglaophyton, which is that intermediate plant. So it's not non-vascular, but it's not quite vascular either. It's kind of an intermediate. It's like making its way towards becoming vascular. We see the return of these terms hydroids and leptoids, which are going to be in the stem and in here. And we did learn during the moss lecture that hydroids and leptoids serve the purpose of like a xylem and phloem. They're just way less efficient, but they're still better than not having anything at all. But even though it has this kind of simple, precursor to a vasculature, more importantly, this Aglaophyton, being an intermediate plant, the sporophyte plant's already way more complex than we saw in the non-vascular plants. So for example, it's much bigger. I mean, it's much, much bigger. It no longer has to be attached to the gametophyte plant. Why not? Well, because the it has the ability to make its own food. So look, the stems are green, photosynthetic stems. So it has the ability to make its own food. And another way that it's more complex than the previous we saw in the non-vascular is, look how many sporangia are on this single sporophyte plant, right? Multiple sporangia on a single sporophyte plant. So that's more complex. But then by the time we get to Cooksonia, which is here on the right, now we officially have xylem and phloem in the stem. Even though it's primitive xylem and phloem, it's still considered xylem and phloem. It's still considered xylem and phloem for the purposes of this class. It's still considered xylem and phloem. So it's officially vascular. It's official, no longer intermediate. But similar to the intermediate one, right? We still don't have any leaves, right? We have photosynthetic stems, still no leaves. Whoops. Still don't have roots, right? We only have rhizoids. But we also have, at the tips, many sporangia per sporophyte plant, right? Many sporangia per sporophyte plant. So that's more complex. Notice this, look at this growth pattern. Grow, oops, wow. Look at this growth pattern, grow, split, grow, split. And even here, grow, split. What is that an example of in terms of growth pattern? Look here, grow, split. Even over here, grow, split. What do we call that pattern of growth? It's one of the more simple patterns of growth, right? Thank you, Miriam. Yes, equal dichotomy, equal dichotomy. The most basic level of growth that the earliest plants have, the most primitive plants have. Okay, great. Whoops. Okay, so then now we're moving through, we're still talking about the increased complexity of sporophyte plant, right? And look, now we are moving our way through some of these plants. By the time we get to Rhiniophyta, we're already seeing different types of growth, right? Like here, this isn't splitting. This is unequal dichotomy as versus the equal we just saw. We're seeing little weird projections, like these little weird spiky things on the stems, right? Still no leaves, but little weird projections, which are called enations. Oh, I should move this and then I could see them, but here, enations. But look here, this is a big leap. When you go here, this one on the left, the Rhiniophyta, still pretty much has sporangia located pretty much on the tips, But by the time you get to, I gotta move this chat box. By the time you get over here to this right, so these are both different species of Rhinia, but this Zosterophyllophylla, you don't need to know the word Zosterophyllophylla. Just know this slide's showing you two things. One, you're having more complex growth patterns. Sorry, three things. One, you're having more complex growth patterns, right? Because we went from only equal dichotomy to now unequal dichotomy as well. You have the appearance of these little spikes on the stem, which are called enations, which we did not have before. And you eventually end up with sporangia that are on the lateral parts of the stem, no longer just at the tips. Yes, there are some still at the tips. But they're also able to grow on the sides of the stem now. See, here's the sporangium. Here's the sporangium. which allows you to increase the number of sporangia per sporophyte plant even more, because you're not limited to the tips anymore. Now you can have it on the sides as well. So we're still talking about increased complexity of the sporophyte plant here. Okay, I mentioned on the previous slide those little spikes, right, which are called enations. And we talked about how the very first leaves, which are called microphyls, evolved from these enations. So I should go back real quick, and I'll use blue since that's apparently the color I use for, is that what I, wait, hold on, clear. Do I use, oh yeah, oh my gosh. Do I use blue? No, I use red. Okay, I'll use red here too. I thought that, like, what is the point of these enations, right? If it's not a leaf, like, what did the enations do for the plant? Why did they evolve them? Why did they put resources into growing these things, right? Like, there's a couple schools of thought, because obviously we weren't around 400 million years ago, so we have to kind of speculate a little bit. But what we think, based on the fossil record, is that these might have served a few different purposes. Here, I'm circling the enations. They may have been a little rigid, so it may have made the plant a little spiky, in that even if they were not super hard spikes, if you were an animal, it would prevent something from eating it, right? Like some prehistoric animal that we don't necessarily know what it would have been. But in general, if you have little things on your plant body, it reduces how many things will want to eat you. So there's that. But it's also thought that since these plants, this one, and all the ones we've seen so far, don't have leaves yet, it's thought that maybe these enations were ways to increase the surface area of the stem to be able to acquire more sunlight, to increase the photosynthetic rate. So that's what we think they did. And this is based on the fossil record, because we can see, no enations, and then enations that start to have something migrating towards them, which we'll see in this next picture, and then eventually microphyls. That's how we think the first leaves came about. So here you go. On the left is a photosynthetic stem with no leaves, obviously, but no enations either, right? Nothing, just a smooth stem, like we saw in like Cooksonia and stuff like that. Eventually you start to get these little spikes, which we call enations. Notice though, one thing to notice, the enations don't have any, this is vascular tissue here, right? This darker color. There's no vascular tissue in the enation, right? But eventually this vascular tissue kind of starts to migrate in. And when that finally occurs, we officially have the first leaves, which are microphyls. Officially. First leaves. Big evolutionary leap to go from no leaves to leaves. Can you even picture like plants today without leaves? So pretty big deal. Remember, this is all review. This is all stuff that we've already covered, right? All stuff that we've already covered, just reviewing, because our next class, we have our first exam. Here we go. So this is microphyls, right? This is a cross section of a microphyll on the bottom. What is a microphyll? Well, it's simple. It meaning it only, in the case here, it only has a single vein, one vein, which is located right here in that cross section, right? One vein. It has an epidermis, which is on the outside. Notice it has a stomatal apparatuses, right? So it's capable of complex gas exchange, right? Here is a stomatal apparatus. It has a mesophyll. These are all tissues, right? Mesophyll, so that's this whole area here, etc. Mesophyll is where your photosynthetic pigments are going to be, where photosynthesis is going to occur. And yes, of course, the vein includes xylem and phloem because we're in the vascular plants. So sporophyte plant getting even more complex, right? Now we have leaves, officially have leaves. We talked about, this is, think way back to the non-vascular plants and how uncomplex the sporophytes were. Now we're in the vascular plants, and the sporophyte plants even have all these different types of tissues in them. That's a huge difference in complexity. I'm not going to draw all the different things again here because I did it when we went through this originally. So you could pull up, I think this is lecture 5 as well. But there's 3 tissue systems in every single part of a plant. In this case, the sporophyte plant, right? 3 tissue systems. A dermal tissue system, which consists of the epidermis. Vascular system, which consists of the xylem and the phloem. And the ground tissue system, which, yes, question? No? Okay. And a ground tissue system that consists of different things depending on where you are in the plant. If you're in the leaf, the ground tissue system is mesophyll. But if you're in the stem or the root, your ground tissue system is cortex. So those are the 3 tissue systems we talked about in lecture 5. This is also something we've talked about in a few different lectures, but particularly in lecture 5, I think we kind of talked about this the most. Clear these drawings. What is a stele? Remember, this is a term we've seen many times already. My favorite topic, right? No, but still an important thing to cover, even though it's not my favorite. So what is a stele, right? We have here protostele, siphonostele with no leaf gaps, siphonostele with leaf gaps, and eustele. They all have stele in them. What is stele? Like here, stele. What is a stele? A stele is a way that how we describe how the xylem and the phloem is organized. The earliest vascular plants, Lycopodium, for example, had a protostele. Here is a protostele. I'm going to draw it. It's a solid tube that in there would have your xylem and your phloem inside of that solid tube. That's a protostele. Slightly more evolved plants, examples Selaginella, have a siphonostele with no leaf gaps. What does that look like? It looks like xylem and phloem are in a ring with a central area that has pith. That's what siphonostele with no leaf gaps. Notice no leaf gaps because we're still only dealing with a microphyll, right? We're still only dealing with a microphyll, so there are no leaf gaps. Once you get to the ferns, which ferns, by the way, are not covered on this exam, right? Ferns are not on this exam, but I'm just, since all of these are on this slide, I'm walking you through the progression. You have a siphonostele with leaf gaps. It still looks like a ring of xylem and phloem with a pith inside. However, depending on where you do the cross section, like if you did it right there, the ring may not be complete because there are these new things called leaf gaps. You can go back to lecture 5 to look at these kind of things. And I think also lectures, well, it's only one lecture, but 6 and 7, right? Also, I think we talk about this. And the most recently evolved plants have a eustele. Eustele is basically, if we were doing a cross section of it, the vascular tissue is in rods. So like here is a rod of vascular tissue. Here is another, I gotta move this. Here is another rod, vascular tissue, right? And remember in the horsetails are weird. in that they have kind of a combo of a siphonosteal and a U-steal. Horsetails are weird like that. You can go to the horsetail lecture to get reminded about that. But why are we talking about this? Because we're still dealing with this increased complexity of the sporophyte plant. Wouldn't you agree that this, one solid tube, is way less complex than this? So that's why we're going through this. This is increasing complexity as you go left to right. And these are how the vasculature is arranged in the sporophyte plant. Okay, here is more increased complexity of the sporophyte plant, right? This is in Lycopodium, or sorry, this is in the lycopods, so it includes Lycopodium and Selaginella. An increase in complexity in growth pattern. Lycopodium has that equal dichotomy, right? Grow, split, grow, split, grow, split, right? Some more advanced members of Lycopodium have unequal dichotomy. So here we grew, and then this one went here, but then look what happened to this one. Whoa, so much more stuff. It's not the same as this over here on the left. And then Selaginella, which is more advanced than the other two here, has a combination of equal and unequal. So like here you see equal, right? Here you see equal. But obviously if you're looking at the stem, if it splits here and this one continues on, that's not equal. So it has a combination. So from left to right here, left to right, you're going from more primitive to more advanced growth patterns. What this is showing us, and let me just see where I'm at with, yeah, I think we're getting there. We're getting there. We'll make it in time. I think this review is really important because it takes all of the little details that we learned in the individual lectures and puts it in this like big picture of plant evolution. So I think it's helpful. It's helpful to put all the puzzle pieces together, right? These are just not random puzzle pieces every class. They all go together in a puzzle of plant evolution. Okay, now we're in horsetails here. Why are we looking at horsetails? Because we're still talking about increased complexity of the sporophyte plant. Some of the ways that horsetails are more complex is they have sporophyte plants that have branches now. Look, right? What are all these things coming off of this? This is branches. We didn't have branches before. In addition to branches, we have some species where there's leaf dimorphism. Sorry, there's branch dimorphism in that some branches are photosynthetic, like here. This is a good one here. This is from the same species, but this branch or this stem is photosynthetic and doesn't make any sporangia, whereas this stem or stem only is not photosynthetic and focuses on making sporangia, right? So we have this dimorphism, right? Dimorphism we've come across many times. Di means two, morph means types. So it has two types of stems, one of which is photosynthetic, one of which just focuses on making spores. The horsetails are also more complex in that now they have megaphylls instead of microphylls, even though, for what it's worth, these leaves don't look like any other leaves we're used to seeing. But I just want to point that out because that is true in terms of increased complexity. But we need to spread our offspring or our progeny to further distances. If we're going to occupy more niches, we have to do that. We have to do that. So sporophyte plants have to evolve additional structures, and they do. And they do. So back in the non-vascular plants, we had sporangia or a capsule kind of thing, right? capsule on the polytrichum, hanging upside down capsule on the liverwort, horn-like looking capsule on the hornwort. Now we're in the lycopods. Sorry, we're in the lycopods, Lycopodiophyta. And now we don't have that anymore. Instead, now we have leaves, because we have leaves now. We have actual leaves now. And they're called microphyls. And some of those leaves have sporangia on them, or a single sporangium, one sporangium, plural sporangia, right? And if you're a microphyll and you have a sporangia on you, we can call you a sporophyll. So here, up here, these are just simple microphylls with no sporangia on them. So we just call those microphylls. These microphylls down here, I should maybe change colors because it's on like a red background. These microphylls down here happen to have a sporangia on them, right? Look, here's the sporangium. Such that now we can call these microphylls, which is right here, sporophyll, because it's a leaf with a sporangia on it, right? And the sporangia is going to make the spores. So we want to spread our offspring to further distances, so we need to make gametophyte plants grow other places farther away. So we have to develop more structures. This is an example of Lycopodium. And sometimes, so this is showing you sometimes the sporophylls look just like the regular microphylls, right? Like in number 1, right? All of these look the same. They're green. Some have sporophylls. Some of them have sporangia on them. Some don't. They all look the same. They're all photosynthetic, and they are not arranged in a strobilus. But when you get to the more advanced members of Lycopodium, as shown here in 2, sometimes you'll see sporophylls looking a little different in that they are smaller. They are not green, which means they're not doing photosynthesis. And they're sometimes organized into a strobilus. Here is a strobilus. Here's a strobilus. Each of the things on this strobilus are sporophylls. They're not green. If you're going to be organized into a strobilus, you have to be small, smaller than if you're not, because you have to fit. You have to fit on this thing. And then in your even more advanced species, sometimes the strobili are elevated on a stalk called a peduncle, right? For your more advanced species, because that makes the strobilus be even higher up so that wind can take the spores even farther. Okay. Right. I think I already said this basically on the previous slides, but again, just really reminding you about going from a more primitive to a more advanced structure. By the time you get to the horsetails, the area of the plant that sheds spores is even more complex in that we have these new structures that only horsetails have called sporangiophores. So this whole thing here is a strobilus. Each of these mushroom-looking things are sporangiophores. Here is one sporangiophore. Here's another sporangiophore. And what's cool about the sporangiophores in terms of being able to spread more progeny, more distances in terms of spores, is like each sporangiophore now has, I don't know, 10, 20, it'll depend on the species, sporangia. Many, many, many sporangia per sporangiophore. Many, many, many sporangia per sporangiophore, as versus one sporangia per sporophyll, right? One sporangia per sporophyll in the lycopods is not as complex as many, 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 like 10 or 20, something like that, sporangia per sporangia 4 on the horsetails. So even more complex. Okay. You have to provide better protection for the egg and ultimately the zygote, right? Because wherever the egg is, it's going to be fertilized by sperm, and that's where the zygote will be, right? Because egg and sperm is the zygote. So over time, we see this not only a size reduction. So if you're going to, let's put it this way, if you're going to protect the egg better, getting smaller is one way to do that, because then you can cover it up with more things if it's smaller. So we see we just covered the trend in increasing complexity of the sporophyte plant. Now we're going to talk a little bit about the size reduction of the gametophyte plant, getting more small. And part of that is going to be to provide better protection for the egg and ultimately the zygote. So, oops. And what this is like a long way of saying we're going to talk about the shift from homospore to heterospore. That's all that's saying. Shift from homospore to heterospore. Okay, what is that? Well, remember, so homosporous, homo means same, and sporous refers to a spore. So if you are homosporous, let's go back. If you're homosporous, right? If you're, whoa. First of all, when we're talking about, even though this says size reduction in the gametophyte plant, right? Size reduction in gametophyte plant, don't forget that when you're talking about homosporous or heterosporous, or here it says homospory versus heterospory, but if you're talking about a plant that demonstrates homospory versus heterospory, you're talking about the sporophyte plant, because the sporophyte plant makes spores. Sporophyte plant makes spores, not the gametophyte plant. But in order to have a reduced gametophyte plant, we need the sporophyte plants to go from producing homosporous spores to producing heterosporous spores. That's how we're going to get to our gametophyte plant being more reduced. So remember, here you go. Let me move this. Let me move that. All the plants, okay, all of the non-vascular plants have homosporous spores, right? Moss, liverwort, hornwort, they all have homosporous spores. When you get to the vascular plants, Lycopodium has homosporous spores. Lycopodium has homosporous spores. And for the record, horsetails also have homosporous spores. When the spores are homosporous, we know certain things. We know certain things. Number one, this is the spore, by the way. Here you go. This is the spore. And the little thing poking out is the gametophyte plant. If you are a homosporous spore, it's like a spore is a spore is a spore. We know then that gametophytes are going to grow outside of the spore wall. So here is the spore, and that big thing coming out of it is the gametophyte plant. So we know they're exosporic, right? The gametophytes are exosporic. They grow exo or outside of the spore if the spores are homosporous. If the spores are homosporous, we also know that the gametophytes are usually bisexual. So here is one gametophyte plant. Notice it's not inside of a spore, right? It's outside of the spore. Spore's gone now, actually. And on it, there is Archegonia and Antheridia, both sex organs on one plant. Monoecious. So we know those things if the spores are homosporous. However, oh yes. Yeah, this is just to remind you when you're looking at these slides later, like, so these are all examples of, we're talking about size reduction of gametophyte plant, and we're talking about species that have homosporous spores. So look, this is the gametophyte plant of the mosses, rather big, right? Rather big. And those species have sporophytes that produce homosporous spores. And look how big the gametophytes are. They're growing outside of the spore wall. They can be big. Get to liverworts and similar, right? This plant sporophyte makes homosperous spores. And so therefore, when the gametophyte grows, it can get big, doesn't have to be inside of a spore, grows outside of the spore. Same thing with the hornworts. They make homosperous spores. So when the spore lands and grows into a gametophyte is outside of the spore wall, right? This green thing is the gametophyte plant. But even looking at those, we see it getting smaller and smaller. We see the gametophyte plant getting smaller and smaller. Look, mosses, green and fluffy, big and green and fluffy. Liverworts, flat green thing, right? Flat green thing. Hornworts, even smaller, flat green thing. There it is, kind of like green blobs. Once you get to the lycopodium, even smaller green blob shown on the left. Don't worry about these. Don't worry about these. Even smaller green blob gametophyte plant. And then some species have a gametophyte plant that almost looks like a little carrot, right? But still super small, still super small. And even in the horsetails, which I also mentioned are homosperous, still a green thing that's growing outside of the spore, but super small still, right? Super small. So we see over time a reduction in the size of the gametophyte plant, even in the homosperous species or phyla. But when you get to Selaginella, officially the gametophyte is really, really reduced, really, really reduced. How is that possible? Because now Selaginella has sporophyte plants that produce heterosporous spores. Hetero meaning different, spores referring to spores. So Selaginella produces two types of spores. Those names of those are microspores and megaspores. Those are the two types of spores. The megaspores are made in a sporangia that only makes megaspores. So we call it a megasporangium. And the microspores are made in a sporangia that only makes microspores, or microsporangium. Because we have these new micro prefixes, we could add those to the actual leaf that bears them. So the leaf that bears this sporangia, remember this sporangia has in it megaspores, so we can call it a megasporangia. And likewise, we can call the leaf that it's on a megasporophyll. This sporangia up here makes microspores, so we can call it a microsporangium. And likewise, we can call the leaf that it's on a microsporophyll, right? The terms micro and megasporophyll will show up in subsequent lectures after the exam. So that's why I'm really pointing them out here. But the main thing that we're concerned about today is this, right? Size reduction of the gametophyte plant. Where is the gametophyte plant now? It's inside the spores. Inside the spores. So super, super small. It's inside of the spore. What's inside of the megaspore? Female gametophyte plant. What's inside of the microspore? Male gametophyte plant. Right? Male gametophyte plant. And the male gametophyte plant is so reduced that now it's just, remember I said Dr. Casper used to call it testicle in space, because now inside of that microspore is just an antheridium. That's it. That's it. Look, that's all that's in there. That's small. Think back to a moss when you're like a big green fluffy gametophyte plant, and now you're here in the heterosporous Selaginella, and your male gametophyte plant is literally one antheridium. Here it is, one antheridium inside of a spore. Spores are heterosporous. Gametophytes grow inside of the spore wall. One antheridium inside of the microspore. Female gametophyte plant. Here she is poking out just a little bit. She's poking out just a little bit outside, but she's inside of this megaspore, right? She has more of a full body than the male gametophyte plant, because the male gametophyte plant is a single antheridium, whereas the female gametophyte plant still has, she's got lots of archegonia. She even has rhizoids. I mean, she's a lot bigger and has a lot more of the things we're used to talking about with the gametophyte plant. But size reduction in gametophyte plants in heterosperous species, gametophytes grow inside of the spore wall. That is important, important, important. Oh, and I got this little guy down here too. Obviously, if each gametophyte plant is inside of its own spore, the gametophytes have only one organ of one sex organ type on it. So they are unisexual or dioecious, right? Two separate houses, in this case, two separate spores. Okay. And looky here. So here you go. This is real picture of megaspore and microspore. Look how big megaspore is, hence why we call it megaspore. Look how small microspores are, hence why we call them microspore. Inside of this all the microspores is going to be one antheridium. Inside of megaspore is going to be a relatively complete female gametophyte plant. Okay, I think that is it. Yes. All right, so we got 10 minutes here, and I just did a crash course review through everything we've covered so far, big picture style, because we've looked at these things in detail in each lecture. And today we kind of put it together to really reaffirm some of those core course concepts. So hopefully by now you've pulled up the study guide, which was posted around 9 a.m. on Tuesday. And so I just want to take this time to give you a chance to ask me anything about anything that is confusing. Maybe there was something on the study guide you weren't sure what I meant, or you weren't sure how much detail I wanted, or something like that. Or maybe you got a question wrong on an assignment and you're not sure why I marked it wrong. So this is a time to ask me those things. I'll give you a couple minutes to kind of get oriented and just kind of look at things and see if you do have any questions. If you don't have any questions, you are free to go for today. The exam is on Tuesday. It starts at 10 a.m. We won't meet in person. We won't meet for lecture on Tuesday. We won't meet. Instead, you go on Blackboard and take the exam. The exam has about 40 questions, something like that. I think about 42 questions. Some of them have, a few of them, like less than 5% of them, have more than one part. So most of them are pretty straightforward, like answer one question, and then out of 42 of them, maybe like 3 or 4 have like 2 parts or something like that. You'll have the entire 80 minutes from 10 a.m. to 11:20 to do the exam. The exam is the exact same format as all of the assignments we've had so far, so there won't be any surprises there. I think that's it. Unless people have questions, I'm happy to answer questions. This is time to ask me. I'm here. Don't be shy.
Professor?
Yes.
Okay, so this is a question from assignment 2.
Okay.
I'm just confused, like why I got it wrong. Because, okay, so the question, it said, some non-vascular plants are early successional. That means that and then we had to answer it. So I had put can grow on other plants because I think you mentioned like that they're epiphytes. They can grow on trees and stuff like other plants. So I got it wrong. I'm a little confused why.
Sure. Here, let me use the chat box, and that might be. Oh, there's a question. Hold on. Here, hold on one second. Let me answer Diksha's question first. So, just so you know, so early successional is one thing, and then epiphytic is a separate thing. So early successional means that you are the first to colonize new habitat. First to colonize new habitat. So that could be, for example, if there's a volcanic explosion and all the land is bare. If you're an early successional plant, you will be one of the first ones to show up after there's nothing there. Another example I gave is like, if you have a new island made by volcanic activity, and then all like like the Hawaiian Islands, right? Like a new island shows up and there's obviously no plants or animals on it yet. If you're early successional, you could be one of the first to show up there. That means that if you're early successional, you can grow on things like a rock or really, really, really bad soil, really bad soil, like not great places. Another example is a mudslide. Let's say there's a mudslide and takes everything away. The plants that first show up are going to be early successional. Whereas epiphytic means that you can grow on other plants. So if you can grow on other plants, we can call you an epiphyte. So some liverworts that we looked at can grow as epiphytes. Selaginella and things like that can grow as epiphytes. So an epiphyte means it grows on other plants. Early successional means first to colonize new habitat. Does that answer your question?
Yeah, thank you.
Yeah, no problem. I know it's kind of like, I think a lot of times in the lectures that early successional and epiphyte show up kind of close together in the slides, so it could be a little confusing. Hira, I think I'm pronouncing your name correctly, Hira. Will there be diagrams? No, there won't be diagrams. Diagrams is something that would be more of like a lab exam type thing. Any other questions of things, maybe things that were unclear on assignments that you got wrong and you don't know why? This is the time I can help you figure it out. I'm happy to. If you don't feel comfortable asking me questions here, that's fine. I do have office hours at noon. So if you want to meet privately for office hours, send me an email and I'll send you a separate link for that. Brittany, do you have a question? Your name popped up on the screen.
Yeah, I'm just confused on so for example, Lycopodium, they have sporangia located on sporophylls, and they have one spore on the sporophylls, and those are homosporous, right?
Correct.
And homosporous, that means, what exactly is homosporous? That they have male and female spores in that one sporangia?
No. So try very hard to not think of spores as male and female, because
Okay.
spores don't have sex. Spores are not sex anything. They are spores. The male and female, if you want to think male and female, think gametophyte plants. Gametophyte plants. That'll help clear things up to begin with. A homosporous just means that if you collected spores from a homosporous species, all the spores are the same. They're all the same. They all just look, they're just a spore that grows into a gametophyte plant once it lands in the ground. So I think what you're asking is, for example, on Lycopodium, which has sporangia on a sporophyll, inside of that sporangia is just one type of spore. And when that sporangia opens and those spores fly out and land somewhere, they will grow into gametophyte plants that have both male and female sex organs on them.
Okay. And then that is very helpful. And then for microsporophylls and megasporophylls, those So a microsporophyll is male, right?
Okay, again,
Damn it, confused.
be careful about, so gametophytes are male and female, or both. So yes, the word micro refers to that it's going to make a male gametophyte, but so on a micro, so on Selaginella, which has heterosporous spores, right? 2 types of spores. You have a microsporophyll that has, so let's put it this way, to simplify things, you still have the same thing. You still have a sporophyll with sporangia on it. You still have that. But some sporangia are only going to make microspores, and some sporangia are only going to make megaspores. Because of that, we can call them a microsporangia and a megasporangia, but they're still ultimately sporangia. It's just that they're making 2 different types of spores, 1 type in each type of sporangia. When the microspores are released, they're not going to grow into anything. They just have, in each microspore, they just have a male gametophyte plant, which is now a single antheridium. And when the megaspores are released, the megaspores contain the female gametophyte plants. And so what ultimately happens is that a microspore will land on a megaspore, and that will allow sperm and egg to come together.
Because the gametophyte plant is inside the spore.
Correct.
Whew, okay.
Correct. Or endosporic, right? Endo meaning inside, right? Yes, correct. Let's see. Andrew here wrote, is there anything you feel that students commonly get confused on that we should review? I think what Brittany just brought up is one of the things that students tend to have some trouble with, like that, because that's one of the more complicated topics of the course, is this idea of like shifting from a homosporous existence to this heterosporous existence. So that can be confusing. And so I think hopefully what I just explained here helped with that a little bit. In terms of that, is there anything you feel that students commonly get confused on that we should review? I think sometimes students have a hard time separating terms that might have similar structure. So for example, sporophyte versus sporangium, or sporangiophore. Like sometimes students see words like that and they just kind of give up before they even try to really understand what they mean. And so then when they encounter them and see these similar looking words, sometimes students have a hard time figuring out which is which. My recommendation for that would be to use the technique that I use to pull things apart. So for example, with like sporophyte, right? We know that sporo equals spores, and we know that phyte equals plant, right? The sporophyte is a plant that makes spores. Whereas another example, because it kind of looks similar, sporangia, right? Has that similar beginning part, so it might look similar to sporophyte, but a sporangia is a structure on a sporophyte plant. Sporangia is part of the sporophyte plant, and a sporangia structure or organ that makes spores, right? Whereas sporangiophore, which we encountered in the horsetails, sporangio refers to spores, and phore, as I've said many times, refers to bear or carry. So it's a thing that bears or carries. Oh, sorry, sporangio refers to sporangium, not spores. Sporangio refers to sporangia, right? So it's a structure that bears or carries sporangia. I think if I'm describing something that sounds like you and you're like, oh man, I just, all these words look the same to me. Pull them apart. Don't just give up. Don't just give up because it looks hard. You can do it. I have faith in you guys. I know you guys are smart. I know you guys are, you're most, I think everyone here is a senior in a biology major. You can do it, okay? You can handle it. Don't get frustrated. Just pull it apart. That's why I do that in the class. Brenda, I think might have, I don't know if Brenda is still here, but she had said gametes are bisexual in homospore. All right. So gametes are always just egg and sperm. Let's be clear here. This is maybe one of those. So gametes equal egg and sperm. Brenda, if you were asking if gametophytes are always bisexual in, what was it? Homospore. Yes, look, homosporous spores, which are from sporophytes, grow into monoecious, I guess you could say bisexual, gametophyte plants that grow outside of the spore. And then heterosporous spores grow into dioecious, might not have spelled that right, unisexual gametophyte plants. They grow inside of the spore. The things I just wrote are also on that slide, on the slides. There's slides here that can contain what I just wrote here. Any other questions? No problem, Andrew. Any other questions?
Professor, this is the last one. Thank you so much.
I'm so happy to help. Yes.
So Salaginella, that sounds cool. They're heterosporous, so both of the gametophyte plants are inside the spore, and they come together, and then the gametophyte plant falls off. But for the lycopodium, I don't totally understand how the gametophyte plant would fall off.
Okay. Let me do this. Because I think I need to put something in order to draw. Here, new slide. Okay. And I guess I need to share. All right, so share screen. And here we go. And share. Okay. Can you see this white slide, like this blank slide, Brittany?
Yes.
Okay. So I think you're getting confused in one way here, which is, I don't know, I don't quite know what you mean by gametophyte plant falls off. I'm not really sure what you mean by that. So can you clarify a little bit what you mean by that?
Like, so in non-vascular plants,
right?
The gametophyte is reduced, so the sporophyte is free living, and then
nope, not in non-vascular plants.
I'm sorry, vascular plants.
Yes, in vascular plants, yes.
Right. So the sporophyte plant is free living, and the gametophyte plant eventually falls off of the sporophyte plant, right?
I see. Okay, I see where the confusion lies. Okay, I see where the confusion lies. Here, okay. First of all, let me ask you this. What does a sporophyte plant make?
Spores.
Right. And so those are what are going to fall off. It's the spores that are going to fall off of the sporophyte plant. if you're homosporous like a lycopodium, so here, look, this is a lycopodium, right? These things I'm drawing are sporophylls. Okay. Here is selaginella. Things I'm drawing are sporophylls as well. Okay. Both of these are sporophyte plants. So both of these make spores. You just told me, right? You just told me that they make spores. And let me write here, lycopodium. Oh wait, I can use text box because it's much better. Text. Lycopodium. Boom. Another text box. Selaginella. Boom. Okay. Both of these are sporophyte plants. Lycopodium, though, like all the other plants we looked at so far, except for selaginella, is homosporous, right? Lycopodium is homosporous, just like all the other ones we looked at. So here is a sporangia. Here is a sporangia. Sorry, a sporangium. Here is a third sporangia, right? This lycopodium has 3 sporangia on it, right? Right, Brittany?
Yes.
Okay. And because lycopodium, like all the other ones we looked at, is homosporous, each of these sporangia are going to release the same exact thing. Here they go. Boom, boom, oop, boom, boom. Here they go. Oh gosh. Get out of here. Get out of here. Get out of here. Get out of here. Let's make them dots. Dot, dot. Here they go. They're released. What are those things that are being released, Brittany?
Those are spores.
Those are spores. Are they different from one another?
No, they're all the same.
All the same, homosporous. And oh, boom, there you go. It landed in the ground, right? Landed in the ground. Now, that spore that landed on the ground is going to grow into a gametophyte plant. Here it is.
Okay.
And because all the spores are the same, and look how big that gametophyte plant is compared to those dots. So is it inside of the dot? No.
It'll be outside the dot.
It grew outside of the dot, right? So we know this gametophyte plant grew outside of the spore wall because look how big it is compared to the spores. And because all these spores are the same, what sex organs are going to be on this gametophyte plant?
Both.
Correct. Both male and female sex organs, because all the spores are identical. So there's no way for the gametophyte plant to have any different information. All right,
Okay.
so that's lycopodium. Now let's do selaginella. So selaginella is heterosporous, right? Not homosporous. It's the only plant we've looked at so far in this course that is heterosporous. Because of that, we know it's going to make 2 different types of spores, right? And because of that, we know that it must have 2 different types of sporangia. So I'm going to use different colors to show those. So here is a sporangia. Here is a sporangia. And because I'm running out of leaves here, because I'm running out of leaves here, I'll use both sides just so you can get an idea. Even though I only use the one side on the other. Here's some sporangia. They're different colors. Why? Because they make different types of spores. Let's say the blue sporangia are microsporangia, meaning they're going to make microspores. Here they go. Here they go, flying out, right? And even if you look at the microsporangia itself under the microscope, it usually kind of has, you can see all the spores, they're all in there, all the microspores, and they're like little and small and whatever. But anyways, here they go, they're flying out. Okay. Brittany, what's inside of those microspores?
Male gametophyte plants.
And what does it consist of now? What is
our antheridium?
How many?
1.
And what else? Anything else?
No.
Right. Just right. Just one antheridium in each of those microspores. Okay, here we go. This is another sporangia on this on the Selaginella, and inside of it, there's these much bigger things. And this is going to be your megasporangia, which is going to release your mega spores. Notice each, here, if I did another one here, each megasporangium can only hold four megaspores, or a tetrad. They're so big, it can only fit 4 total megaspores, or 1 tetrad. So here they go. Megaspores are leaving. Here they go. Here's another one. Another one. They're all leaving. Brittany, what's inside of those megaspores?
I believe you said they have multiple. Well, okay, it's one female gametophyte plant.
In each one, right?
Yes.
Okay. And so the female gametophyte plants have kind of the normal stuff we're used to. They have many archegonia. They even have rhizoids that stick out a little bit. And so are these gametophyte plants growing outside of the spore?
No, they're growing inside.
Correct. So here's what's going to happen. A megaspore will land on the ground. And if she's lucky, a microspore will land. Usually a few do. But the point is, if she's lucky, a microspore will land on her. If that happens, the sperm from the microspore can then fertilize the egg that is inside. So let me be more clear. The microspore contains a male gametophyte plant. The male gametophyte plant makes sperm. If the microspore that contains that gametophyte plant lands on the megaspore, and the megaspore contains a female gametophyte plant with an egg, the sperm from inside of the microspore, because that's where the male gametophyte plant is, can leave and fertilize the egg of the female gametophyte plant who's in that megaspore. And then when that happens, eventually so here, when that happens, sperm and egg fuse, and then you grow a new Selaginella sporophyte. Does that make sense?
That is so clear. I don't know why this wasn't clicking for
No,
me.
it's okay. And likewise on the left here, on the Lycopodium gametophyte plant, that's the darker green. Remember that grows outside of the spore wall, that has male and female sex organs on it. And if the egg and sperm come together, you'll eventually have your Lycopodium sporophyte plant emerge from that. And because the sporophyte is dominant in both of these things, eventually the gametophyte plant, this is dirt, eventually the gametophyte plant's just going to go away because sporophyte plant doesn't need it, right? And likewise, eventually this will just decompose because the sporophyte plants are independent and they don't need it anymore.
This was very, very clear and helpful. Thank you so much.
My pleasure. And if you think of anything else and have questions, I am available via email, and I do technically have office hours starting at noon. So if anybody wants to meet me at noon, just send me an email as well.
Have a good day. Thank you.
Thank you guys. Have a good lab tomorrow.